But I got to tell you that AGT just has way too much for me to go through, data like this. Let me give you the top level, and then I'm here, and we have somebody here from business development. If you hand us a card, I'll send you a 45-slide deck with all of our data in it. You can read it yourself. It's fascinating. It'll prove everything I'm about to tell to you. But I think the critical thing is that we all get on the same page about gene and cell therapy and what AGT is, and that would help you to decide whether we are somebody that you want to engage with. So we're here at Precision Medicine, and basically there's a lot of stuff going on with big data and genomics and diagnostics and a whole bunch of things that are going to get us much more accurate diagnoses and also reveal a lot of the pathology of disease. And that's really where AGT picks up. We're not about diagnosing things, we're about curing them. And you're probably aware that there's about 6,000 to 7,000 monogenic diseases out there. So a disease where one gene is involved in either not making a protein or enzyme that is required in the body, and this manifests itself as a disease. They're all rare diseases, but they add up. The 6,000 to 7,000 monogenic diseases are $2 to $4 trillion of palliative care in the healthcare market. So, it's a huge market, but the question is can you efficiently go out there and start creating cures for all of these different monogenic diseases when some of them are as small as 400 people on the planet? And the answer is yes, but you've got to be incredibly efficient about it. And how do you do that? Well, the way that AGT did it was we spent about 6 or 7 years just researching viral vectors. We're vector agnostic, although we normally use lentiviral vectors. We've even made modifications to lentiviral vectors that make them temporarily expressing as opposed to permanently expressing. But really that was the focus of all of that research. The research was about creating a platform where we had a huge amount of capability, tools, and components that we could mix and match to cure a myriad of diseases, and to do that in a way where we could actually go into all of these rare diseases. Now, it turns out that it's not just rare diseases that you can cure with a platform like that. And our main product is a cure for cancer, and I kid you not. We have a method for treating epithelial solid tumors. It's an immuno-oncology approach, completely unique, which I'll describe in a minute, that is getting 85% complete remission in mouse models with abscopal effect. So since I have so many PhDs in the room, you probably understand exactly what I just said. But for everybody else, what that means is that I can treat one tumor in your body. Let's say you come to me with any epithelial solid tumor. Let's say you have a lung tumor. I can go ahead and do an intratumoral injection on that tumor with one of our lentiviral products carrying one of our proprietary constructs, which will cause some of the cells in that tumor, not all of them, about 20% of them, to start secreting a stimulatory small molecule. For the PhDs in the room, that is IPP. It is a 5-carbon pyrophosphate molecule. And what does that do? Well, what we discovered is that the TCR on a type of T cell that you have in your body, everybody understands that immunotherapeutics is about activating T cells against the target, right? And there's a lot of ways to do it. We think the safest way to do it is not to modify the immune system to be hyperactivated against something. It's to modify something you want killed, like cancer, so that it's infinitely attractive to the natural immune system. This is intuitively much safer. Now, 3 to 5% of your circulating T cells are a special type of T cell. It's called a B delta 2 gamma delta T cell. And why do you have so many of them? Remember that your immune system is full of T cells that are ready for anything. There are antigen receptors on some of your T cells that are prepared to capture viruses that we haven't seen on the planet in 2,000 years. So you have almost an infinite diversity of T cells, and yet 3 to 5% of them, all they do is circulate around your body and eliminate small occurrences of epithelial malignancies. And this is the only reason that you're sitting here in this room listening to me today. If you didn't have gamma delta T cells, you would have been dead a long time ago. You'd last about a year without them before you had solid tumors all over your body. So it stops it early and it doesn't grow into a solid tumor. But what about when you just, some random combination of factors ends up getting a critical mass of these cells together, and it starts growing like a balloon and the surface area is too big and these gamma delta T cells can't eat it fast enough? That's when you need radiation, surgery, and chemotherapy. Well, in fact, we think in the future you won't need that. And if the mouse experiments translate to the humans, which I think they will, I think they're going to translate even better, and I'll get back to that if I have time, then you won't need that anymore. Because if you come in with any epithelial solid tumor, I'll take about $30,000 worth of lentiviral vector. I'll inject it intratumorally into that tumor. So let's say it's a lung tumor. It'll start secreting that IPP. That stimulates the TCR on those gamma delta T cells, but they don't target using their TCR. They target using an NK receptor. The NK receptor is exquisitely specific to malignancy. So these things activate, proliferate, and they will start attacking the all of the cells that are in there, measuring them with their NK receptor, and if the NK receptor matches the stress molecules that are natural on any one of these malignancies, it'll activate and kill that cell. But it's killing them at 300 to 600 times the normal rate. And this is enough to clear that solid tumor. Now remember, it's not targeting those 20% of cells that are secreting the IPP. No, they're just activating and they're eating and querying. They're in hunt and kill mode and they're clearing things faster. And so what you get is all of the malignant cells go. It creates a catalytic event in the tumor microenvironment that causes this activation, proliferation of those T cells, and it clears every single malignant cell in that tumor, whether it's secreting IPP or not. And here's the side effect. These things circulate around your body. And when they do that on their way to the lymph nodes and replicating or just circulating around the lymph and the blood system, what are they passing? Secondary tumors and metastases. And we're seeing that those are getting cleared in the mouse models as well without any treatment to those tumors. Now, why will this work better in humans? Because you have gamma delta T cells. Mice don't even have them. We literally, to do these experiments, we put different types of cancers around the mouse's body, and then we put gamma delta T cells from a human into the peritoneum. So we can treat a liver cancer tumor on one flank, put a breast cancer tumor on the other flank, treat the liver cancer tumor, inject the gamma delta T cells, and both tumors go away. Of course, you're still going to someday get another critical mass of these malignant cells growing together. You may get another solid tumor, and you'll need another shot. This is the nice thing about lentiviral vectors. We can use the same drug on you many times. So we would be able to basically wind back your cancer age by 5 or 10 years, and if it reoccurred, you'd come back in for another shot. No radiation, no chemotherapy, no surgery. As a matter of fact, you cannot remove the primary tumor. Surgery would actually be ineffective. We need the primary tumor to create a vaccine effect in your whole body. The mouse results really show that. We're collaborating with Stanford Medical School, Dean Felcher. He's a worldwide recognized expert in hepatocellular carcinoma, which is one of the most deadly liver cancers, the most deadly liver cancer. We think we'll get a proof of concept in 2020 in his clinic. So that is really, I think, AGT's biggest, most exciting product. Of course, we have monogenic cures, just like I told you earlier. So one of them is phenylketonuria. It's within the therapeutic index of our current drug candidate to cure phenylketonuria, even in neonates. There's a lot of advantages to lentiviral vector there as well. It's a one-and-done cure, and we're working on that with the University of Pittsburgh Medical Center, Jerry Vockley. We think that that will go into a pre-IND meeting this year. We're talking with a number of different companies about out-licensing that because that one's getting squeezed between two things. All of our scientists are going into immuno-oncology, and all our money is going into a cure for HIV. This year, we will enter the clinic with a one-and-done cure for HIV. And the way that works is we take a 400 milliliter leukopak from you, we amplify the T cells that clear your body naturally of HIV. Those are HIV-specific T cells. This is the problem with the old approach of doing this. Everybody thought, just strip CCR5 from all your T cells and you'll be immune. The Berlin patient, voila, it's so simple, even a non-scientist could think of it. But here's where the scientists were critically important, the PhDs. What they recognized is what makes you immune to HIV is the HIV-specific T cells. And if you have critical mass of HIV-specific CD4 positive helper T cells, they'll make a completely diverse reaction to HIV virons that will even catch all of the mutations. And that if we go ahead and take your blood after you've been treated on antiretroviral therapy, so if you had HIV, you go on antiretroviral therapy for 1 to 3 years, you get those HIV-specific T cells back. If we remove your antiretroviral therapy, you viral rebound in 21 days because those things are the first T cells that get depleted in your body, are the HIV-specific ones. Why? They run to the infection. Then they get co-opted by the viron, invaded. Those are the only ones we have to protect to make you immune. We know this because we've done this on the patient blood of 8 patients, and we have blood now in the lab that doesn't just sterilize itself of HIV. You can pour as much HIV blood in there as you want. It sterilizes that too. This is tremendous. You can see this in the data deck. This is something that we're going to come out with this year. We're going to test this at University of Maryland. The people that are associated with this project are also in the deck. It's the who's who of HIV. My confidence level is tremendously high about that product. We are returning memory effect of the critical T cells protected against HIV infection to the body of a patient. They no longer need antiretroviral therapy. They cannot get AIDS. They can no longer infect anybody. And here's the best part. They can never be reinfected. Permanent immunity. Anyway, thank you for your attention.